So, hey everybody, I'd love to introduce Jim Tucker, Pierre, Hiernaux, and Ankit Kariryaa from, I guess, when you wrote the paper, it was NASA Goddard, right?
Yes.
And talking about, well, looking at forests from space. Welcome.
Thanks. I'll be going through some slides from a recent paper of ours in Nature from early last month. And the four principal authors are the first four authors on our paper: myself, Martin Brandt, who unfortunately could not join us, Pierre, Hiernaux, and Ankit. And we indicate in the paper that the four of us should be considered the first author on the paper because of each person's unique contributions to what turned out to be a fairly broad paper, as well as a fairly deep paper. So what we did was we used a large volume of satellite data, if I could have the next slide, Ankit, to convert tree crowns over an area as large as the area of the continental United States and Alaska, about 10 million square kilometers, from the Atlantic Ocean to the Red Sea, north of the Equator and south of the Sahara. We used 1/3 Maxar images to use machine learning. The learning part of our study was 89,000 training data of individual trees. We burned a lot of HPC, of high performance computing time, on the data we organized for a matrix of 2 × 10⁻13 elements. And after 60 million core hours on blue waters before it was decommissioned, we mapped actually 13 billion trees over the entire area and 10 billion trees with tree crowns greater than 3 square meters in the semi-arid portion of our study, which we converted into carbon, totaling 0.8 petabytes of carbon, disaggregated into leaf, wood, and root carbon at the individual tree level. Anytime you do a study like this, you put yourself in a box. How do people use your data? We address that, and we have a viewer associated with our paper which allows you to look at our entire data set of the 10 billion trees, location, and carbon at the tree, down to the tree level. Next slide. So our study took part on the south side of the Sahara, and we used Maxar satellite data in the early dry season. The top image is the rainy season, where everything is green. The bottom image is the same area. You see the same trees. You see all the herbaceous vegetation is senescent, and we focus on the tree crowns. Next slide, please. Here's the same picture with an NDVI profile of this area in Senegal, which you see in the middle where the pointer is now. What we have is a single rainy season. It is very pronounced. You have the dry season, then you have a rainy season of 3 or 4 months, 5 months in some locations, depending upon latitude, and then you go into the dry season. All of our data, which we use, are from October to March during the dry season. Next slide.
Question. Why? Are tree crowns more pronounced in that season?
Yes. So we want to work. Well, there are 2 reasons. One is cloud cover is significantly less. And secondly, we have a pronounced difference between trees and everything else. As you can see in this image, in the rainy season, the herbaceous vegetation is green or actively growing. In the dry season, in the bottom image, the herbaceous vegetation is senescent. So we picked months of maximum contrast between tree crowns and their background.
Thank you.
So let's go to the next slide, Ankit. Thank you. So this is how things look for our training data. This is and also what we mapped. The arrows on the right point to the same tree, which is on the left, and we use the panchromatic band to identify the shadow. And our definition of a tree is it has to have a green leaf canopy, and that's indicated in the bottom image with the normalized difference vegetation index for this specific tree, and an associated shadow in conjunction with the green tree canopy. This is what all of our training data are based on under a wide variety of conditions, viewing angles, aerosol optical thicknesses, and some under different cloud cover scenarios. And you can see how the green tree crown is separated from the shadow, but contiguous to it. Next slide. Here's one example of one tree on the left-hand side of the image, and the same tree as we have processed our data with a convoluted neural net UNet architecture and identified that tree. And we have not identified as trees any of the shrubs and grassy areas, either because they are dormant or because if they're green, they do not cast a shadow. So next slide, please. So from our study, we identified 9.9 billion tree crowns greater than 3 square meters of area for the area we show here which goes, excuse me, which has a precipitation range from 0 millimeters per year in the far north of the image down to the bottom of the image, which has 1,000 millimeters of precipitation per year, and then converted each of those 10 billion trees into carbon at the tree level. Next slide. This is a picture of me taken, God forbid, 42 years ago in Senegal. I started working in sub-Saharan Africa in the early 80s using satellite data. This is where we at NASA developed time series remote sensing using data from meteorological satellites. This is where I met Pierre, Hiernaux, who will follow Ankit in the program, and just a picture of me in the same area during the rainy season. So this is an area where Pierre and I have worked. Pierre has extensive experience. I have an episodic series of experiences there. Next slide. Now, when you form a data set of 10 billion trees and convert them into carbon, you can look at different distributions of what you have. You certainly have a huge sample size. So if people ask you, what's your sample size? In our case, we can say N equals 9.9 billion trees. And on the left, you see a distribution in megagrams of carbon per hectare with respect to rainfall. If you multiply these values times 1,000, you convert the values into kilograms of carbon. On the right-hand side, you see data summarized by individual tree, and you see again that if you multiply the megagrams of carbon times 1,000, you get the kilograms of carbon from wood, root, and leaf from each tree in question, and you see the distribution of these with various percentiles. These are figures from our paper in Nature, and it's important to look at the mean as well as distributions as you see here. Next slide, Ankit.
Question on this one. So the plot shows for different climate zones, it seems like you're ignoring the tree species, because I'm guessing you can't really figure that out. And you're saying that whenever it's rainier, these trees in that climate accumulate more mass. Is that right?
Well, Pierre Hiernaux will go into the botanical distribution of trees in detail. But yes, that's what we're saying, that we, because of our locale, you have a limited species diversity, and we are able to pool our allometry with respect to species. If we didn't have the same distribution of trees, are the same distribution of tree species from the Atlantic Ocean to the Red Sea. You couldn't do that, but we do, and we have, and these are the results.
Okay, and then you were getting roots, but this, are you getting anything about the soil beyond the tree roots?
No, we don't care about the soil. We have enough problems to worry about in terms of roots, wood, and leaves.
Fair. Yeah. Okay, cool. Thank you.
You're welcome. Okay, now this is a plot of the carbon probability distribution on the left-hand side and a distribution of tree crown area in a cumulative fashion on the right-hand side. I'll draw your attention on the right-hand side to the arrow pointing to a 50% point at the 15 square meter tree crown area. Half of our trees are small and have a tree canopy from 3 square meters to 15 square meters, and then 50% have more. So this is why it's imperative to use 50 centimeter data. Otherwise, you miss a huge number of trees which contain a lot of carbon. On the left-hand side, I draw your attention to the top 2 portions which show in the red numbers the percentage of carbon in the hyper-arid, arid, semi-arid, and sub-humid areas respectively. And below them, and below the arrows defining the range for hyper-arid and all of the 4 zones with respect to annual precipitation, you see the area which they comprise. So the point here is 90% of our carbon comes from only 15% or from 30% of our area. And as we go to higher rainfall amounts, 15% of our carbon comes, or rather 64% of our carbon comes from 15% of the area, which has from 600 to 1,000 millimeters per year. You see the sample size, you see the area from which these data come, and you see our total carbon value when you sum everything is 0.8 petagrams. Next slide. We were very curious to compare our results, scaling up from the tree level to what other investigators had reported in terms of megagrams of carbon for above ground trees. This would be the wood and the leaf portion of what we measured, and excluding roots. On the left-hand side, we see comparisons to 6 previous studies using fairly coarse resolution satellite data down to 30 meters by 30 meters from the Landsat satellites. And you notice our data are the black dashed lines, and the gray zone around the black dashed lines of our data represents plus or minus 1 standard deviation. On the right-hand side, you just see a simple bar graph summarizing the various studies with the amount of total carbon, which they estimate. We found only 1 previous study was close to what we had estimated, and that was Niall Hannon in purple, and it followed our plot at least up on the left-hand side to 800 millimeters per year, and then diverged from our data there. So these comparisons reflect the fact that you need 50 centimeter data to measure discrete and scattered trees. If you use coarse resolution satellite data, you're going to miss a lot of the trees. If you use planet data at 3 to 4 meter spatial resolution, you're going to miss a large number of trees, about half of the total trees in the area, because they are small. Next slide. Now this is a different plot on the right-hand side, but the same plot we saw previously on the left-hand side. And this is what happens when you have a huge data set. You can plot things, and you see in the first 4 plots, you see the overestimation of those 4 investigators Um, Actually, the first 5 show an overestimation by the 4 other or by the 5 other investigators, and only Niall Hannon is slightly on the underside of what we have. And because these sample sizes are so large, it makes it very difficult when you use simple statistics like an R value or R square value to express how well these data agree, because we're working with a study area over 10 million square kilometers. Next slide. We also made comparisons to numerical simulation models, and not surprisingly, numerical simulation models work at a very coarse spatial resolution. There are a few which operate at a spatial scale of kilometers. Most are at tens of kilometer scale. And if you don't represent the tree cover as it is, you're going to have very different results from what we have. And so that is shown in C on the right-hand side. And the standard deviation of all of the TRENDY models represented in the right-hand plot appear in gray. And the reason for this wide variation is that previous studies have not used, or previous modeling studies have not used accurate representations of the tree cover. Next slide.
So on that one, what kind of models are these? I mean, I know it's kilometer resolution, but what kind of dynamic do they capture?
Well, they are, they simulate carbon departing from assumed distributions of vegetation, and then they follow this through time. So here we're looking at a summary of this to see how well they agree in aggregate with what we have done with respect to rainfall.
Okay, so what I'm really going for now is you have a study that has a particular estimate in a particular point in time. And now we can do this from, now that we have satellites that are good enough from now on. How would you parameterize those models to take advantage of your data?
That would be done very easily. Now, we are about to start processing these results. This was the first time we had done this. And we did it with 326,000 Maxar images. We're now ready to do it with 800,000 Maxar images. And so therefore we can have really good data for an earlier time period and a later time period. Those data would be very useful for the simulation modelers. The data represented here are distributed over a almost 20-year period, because we had to use what data were available. We didn't buy the data, or else we would have bankrupted everyone who works in NASA and Earth science. But now we're ready to do this in a way which will be much more useful to the simulation modelers of carbon in vegetation.
Okay. And now in terms of the precision of the models, it sounds like these are coarse things that just say the aggregate vegetation grows at this % a year or something rough like that. But your data should be able to go tree by tree, species by species, agent-based kind of a thing, right? It should enable finer grain models.
Yes, exactly. And this is one of the reasons we did this.
Okay, fantastic. Thank you.
You're welcome. Next slide. Now, this is the output data which we have. And so we start with tree crowns, which you see at the top, and they are the green blobs in the top panel, which is labeled tree crown area, on the landscape. We then convert those tree crowns into tree carbon, and you see the color-coded trees expressed in their carbon density. We then aggregate these data to the hectare level, or 100 meters by 100 meters. And that's where you see the checkerboard pattern. And then we combine the tree carbon and the hectare carbon, and you see the bottom panel. So this is why high resolution data are the only way to do this accurately. One, because you can take into account the smaller trees, which in some areas are very important. as well as the larger trees, and then aggregate those data to the hectare scale, which will be more useful for many people, especially the modelers. Next slide. Now, this just summarizes in the top two panels in A and B the data we started with. So we started with 326,000 Maxar images. They covered an area of 90 million square kilometers. You see on the left-hand side of the image on top, the distribution of these images by years. Most of our data come from 2010 through 2020. And then in the bottom panels, you see what data we selected to minimize certain effects we wanted to avoid. So we then stressed the early dry season months of November, December, and January. And we also minimized off-nadir viewing. We minimized cloud cover in the images. We minimized aerosol optical depth. And this was to avoid several contaminants in our data. We were not completely successful with respect to aerosols and clouds, but we will show what those uncertainties are. So this is what we do. We start with a huge volume of data. The more data we have, the better we can do, because we have more opportunities for having better data by a selective process in the formation of the data which we process. Next slide. Now, some of our problems. We have problems with aerosol optical depth, primarily dust storms, which come out of the Sahara. Here's one slide here. We also have problems with residual clouds, which we miss in our initial screening, and you can see the clouds here. So this is an example of that. And if you look at the dust storm, you notice a front in the lower portion of the image. These are very impressive dust storms when they roll across the countryside. Next slide, please. Here's a picture of Pierre Hiernaux on the right with two of his co-workers who work with allometry and botanical aspects of vegetation in this area in a high aerosol optical depth condition in Mali. And one of the features when the aerosol optical depth from dust gets very high is you have no direct light. All the light is diffuse. And Pierre is properly dressed, as you see. He's in an area which is very sandy, so you don't have to wear shoes. If you did, they would get full of sand and in one of these very high aerosol optical depth conditions. Next slide. Now, we had a thorough examination of aerosol optical depth as well as cloud cover. I took part in the cloud cover assessment of the data for every scene we used, and we determined that aerosol optical depth greater than 0.6 affected 3.7% of our study area, and visually determined cloud cover affected 5.3%, and 9% or 1% of our data did not process for various reasons. Perhaps there were corrupted data in some parts of the form data. Anyway, 10% of our data were suspect. Nevertheless, we mapped 700 million tree crowns in the aerosol and cloud cover affected areas. And we conclude from that our aerosol and cloud effects were well within our overall uncertainty of plus or minus 20%. So we looked at these two components in detail and were very surprised that we still effectively mapped 700 million tree crowns. We attribute this to the power of convolution as we employed it using Ankit's code. And we were puzzled at first, but closer inspection showed this was a case of overcoming many conditions of faint high cirrus or aerosol optical depths. In the next run, we will increase the threshold for aerosol optical depth and do a better job of subsequent cloud cover screening before we process data. Next slide.
A question. So aerosol optical depth, does that just change the color, or is that a proper cover of the land?
Well, what it does is it relates to the downwelling solar flux and how much of it is direct or diffuse. And you saw one example under an aerosol optical depth of probably 1.5 there, where all the light, if you notice, there's no shadow which is cast. And so aerosol optical depth determines the degree to which the downwelling solar flux is scattered. Here's a condition where you see in the image now where it is completely scattered and there is no direct component to the downwelling solar flux. We will set our aerosol optical depth at a higher value because we were still able to map several hundreds of millions of trees in an aerosol optical depth greater than 0.6. So we will move that part of our study to a higher value so we eliminate some of these conditions.
Okay, thank you.
You're welcome. Next slide. Okay, now we come, you put yourself in a box when you use convolution as we have to map all of the trees. And so we developed a viewer. So now Ankit will go through one or two slides of the viewer, and then I'll pass the baton to Ankit to talk about many of the computer science aspects of our work, which are really interesting, but frequently we don't get to them because we have so many interesting aspects of the data. So let's go to the next slide. Okay, Ankit will now take over from here and take it away, Ankit.
Thank you, Jim. So now we come to the other part of the study, like basically how do we train our models and what kind of models are they and so on. And before I go into the models, I would like to start with the training data. So the training data for this particular study comes from UTM zone 28 and 29. As Jim already mentioned, we hand labeled close to 90,000 trees across this area, and you can see the distribution of where we hand label the trees in the red boxes in this particular image. So what you'll notice is we have quite a few training sites in the lower part and a bit less in the northern part. There's a huge variety or like huge difference in the tree density in the hyper-arid areas compared to the sub-humid areas. So during our annotation, we have to count for that. So overall, we pose our problem or our learning task as binary semantic segmentation of trees, meaning that given a satellite image, we want to, at each pixel, we want to say whether there is a tree or not a tree and what's the probability of that. And as you can see here, we have hyper-arid areas, arid areas, semi-arid, and sub-humid areas. So there's quite a bit of difference in density of trees. So in hyper-arid areas, you may have huge patches where there are close to 0 trees. And whereas in sub-humid, you may have a complete dense canopy. So there's quite a large class imbalance in the north compared to the south. And we have to basically solve two problems here. First is to tackle the class imbalance, and then the second problem was to tackle the merging of various canopies. And how we do that is first we use Tarowski loss, which helps us tackle the class imbalance better. And in the Tarowski loss, we include a extra penalty close to the boundaries, and I will show that in a second. And overall, this is what our loss formation looks like. And besides that, we use ADDELTA as our optimizer. We start from a learning rate, very high learning rate of 1, relatively high learning rate of 1, and we gradually decrease as each time our model starts to plateau. And then we select the weight configuration with the lowest validation loss as our final model. And what's special about our training procedure is that we take a active learning approach. So basically, we see in what kind of conditions the model is having troubles or having problems, and in there we create new training data. So when we created the training data, that was the approach. And what we would do is we would draw random rectangles, random shaped rectangles. So like this red rectangle, nearly a square that you see here, but it could be any shape. And essentially in that rectangle, this is what defines our training area. We would label all the trees. And then during training, from this area, we would sample 256 cross 256 pixel patches randomly from this area, as you can see. And each patch will be normalized by the statistics of the area from which it is picked up. And we additionally normalize 40% of the patches with its own statistics. So that was our normalization.
I mean, with statistics, do you mean like the tree count or some other attributes?
No, it's simply the mean and the standard deviation of the per pixel values.
Of the pixel values. Oh, so it's a color balance.
Right.
Okay, thank you.
Yeah. And the reason for that is like, as Jim already mentioned, in close to 10% of our data, we have high aerosol density and we have clouds. Also, as Jim already mentioned, we have data from multiple years. And some are from early dry season and some are from late dry season. So we have quite a bit of seasonal and sensor variation. And the data comes from multiple satellites. And with all these differences in the data, what we found was that per patch normalization helped quite a bit because that helped us to overcome many of these discrepancies and basically have better performance in burned or cloudy areas. So that was a reason of normalizing some of the patches with their own mean and standard deviation in the pixel space. And then to basically separate merged canopies, what we do is we add an extra penalty weight in our loss function. And how we do that is using this trick developed by Ronneberger. So if we have closed canopies, next to the borders of the merged canopies, we add extra weight. And you can see the weight map around here. So if this is the hand labeled trees, then from this data we derived a weight mask where we get extra weight on the canopies that are close to each other. And this is what it finally looks like. So we only add extra weight on the border to like canopies that are very close and about to merge and not in other places. And that helps quite a bit in separating individual canopies when you have a merging.
Question.
Yeah.
I've been wondering about this for most of the talk, how you do canopies. So is this manual analysis for like an edge border detection that you just scrape manually as an input to the neural net? Or is this a separate objective for the neural net to train that helps it do the main task?
So this is the weight used here in the loss function. So basically, during training, we pass the weight to the loss function for each patch. Our loss function gets what's the weight at each pixel, and it's 1 everywhere except in these border areas which merge in. So where you have canopies.
So you compute these weights. Are you
Yeah, we compute these weights when we create the annotation mask.
It is not learned. In other words,
it's not learned. No.
Got it. Okay, so now, what are the limits of this? Because I can imagine you go farther south where it's jungle, or like South America, it's all single canopy. Is there hope there?
So we have to remember we are working in drylands. So in mostly drylands area. So in drylands, most of the trees, they grow far apart.
Right.
Because of the scarcity of resources. So that's why this approach works fairly well for drylands. And it does help when you go further south. So we did another study in Rwanda, and there we also use a similar approach. But of course, even with a very high penalty in merged canopies, there you get a lot of canopies merging in. And then you can actually separate them later on, assuming symmetry in the tree crowns, for example. or using other techniques, or you can also use an energy-based model, for example, a deep watershed transform to do this better. So there are multiple ways of overcoming this problem, but for this particular project, what we found, just having a simple binary segmentation with a weight map worked fairly well, and that's why we stuck with it.
All right, thank you.
So as I mentioned, we treated as a binary segmentation problem, and here it's fully supervised. And given an input image, what we want to do is predict tree crowns at each pixel. And for this, we use the unit architecture developed by Ronneberger et al. And we do make some modifications which are fairly common or already in existing literature. So in some ways, we follow what's established for semantic segmentation, and we use it for our problem. Now coming to the other part, like how do we basically evaluate our results? So there are three different ways in which we do our evaluation, and there are three different data sets in a way. So the first evaluation is a randomly distributed, so independently and identically distributed patches in our study area. And in what we did was we selected 78 random plots of 128 cross 128 meters, that is 256 cross 256 pixel. And then one of our team members manually hand labeled all trees in there. So that's our first validation set. So in there we have 5,925 expert labeled trees. And then we also compare with field campaigns in Senegal, where we had measured characteristic of 102 trees. And what you can see in the plots over here is like at a plot level, what is the determination of coefficient between our predicted canopy area and the label canopy area, as well as the prediction density and the label density. So what's the R square between these quantities? And in plot D, you can see over here how does our mapping compare with the field campaign. The reason we are doing it at the field level, especially for this particular part, is that we didn't have the precise geolocation. So we knew we had information about the trees in a plot, but we did not have the location of individual trees. So that's why we are aggregating our statistics over the plot and then doing analysis. So that's one way we are doing our analysis. Just to expand on this a bit. So from hand labeled trees, we have 4,925 trees with a total canopy area of 118,000 square meters. And in the predicted trees, we get 5,915 trees with a total canopy area of 121,000 square meters. And RMSE is around 10 square meters. And our sensitivity and specificity both were close to 96% for the study. So that was one part of the analysis we did, evaluation we did, but we also did another evaluation on top. And here, basically, what we did was, instead of creating hand labels, we took the predictions of the network, and then we went over the predictions, and we basically counted the number of commissions and number of omissions in those predictions. And for this, we considered areas containing 50,000 trees, and we found that over these areas, our total commission rate was 4.9% and our omission rate was 2.7%. And most of the errors actually occurred in the smaller classes. So even if we were over predicting or under predicting, then it was mostly in the 3 to 15 square meters class where we had the most errors. And and essentially what happens is, if you have an area like this with lots of bushes, then many of the bushes will be predicted as trees and vice versa. So some of the trees will not be predicted, and many of the bushes will be predicted. And for a human, it's relatively straightforward to judge because bushes usually don't cast a shadow, whereas trees do. And this you can see, for example, here you have a tree casting a very clear shadow, but that's not the case for most of the bushes. So what you see here in so as you can see in white, and so the circle areas in white and red are trees with high NDVI plus associated shadows, and in yellows we have bushes where their shadows are distributed and not cast in the direction of the solar flux. So that's how we did this third part of the evaluation on for our data. So now coming to what is the uncertainty of our whole approach? And I would like to basically start with a simple statement that we are predicting tree biomass from crown area, and this is inherently uncertain. At the level of individual trees, this is inherently uncertain. So what we are trying to aim for is that over the large area, we don't have high uncertainty, but for a single tree, this is definitely a difficult task and inherently uncertain. So here, what we do is, for example, Pierre will go into the allometry in a moment, and I'm just explaining how we are calculating the uncertainty for the allometry before Pierre mentions that. So what we are doing is we are looking for the absolute and the relative errors in our estimates. And for example, we have our the mean root biomass or the mean root carbon given by this, and then we have our predicted biomass. And what we are essentially interested in, what is the absolute error in these estimates and what is the relative error in these estimates. So we calculate our absolute errors for different allometric equations, and from that we calculate our relative errors. And overall for allometry, our relative error is close to 19%. And we do something similar for our biomass. So essentially what we do is we take our hand-labeled predictions and we take our neural net predictions, and we calculate the biomass or the carbon from both the quantities, and we calculate the absolute error and the relative error from them. And once we have that, we combine the two uncertainties to get a final uncertainty of approximately 19.75%. So yeah, and now Pierre will last slide. Yeah, so as a word of caution, there are a lot of biases that we are just starting to understand. For example, how we measure tree in a field is quite different than how you measure it from a satellite. So Pierre, who's the expert, will explain it better. But in short, in field, what you're doing is for every tree, you're finding 2 points which are furthest apart on the canopy, and then 2 points which are perpendicular. So this gives you the major axis. And then you find the minor axis as 2 points which are furthest apart, perpendicular to the major axis. And then you assume the tree to be in ellipse, and you calculate its area. In the satellite image, we can draw an exact polygon for the tree, which may look like this, for example, a simplified polygon. And then we can calculate the area of the polygon. And just because how you measure the tree on the ground in the field versus how you measure it on the satellite, it also creates some bias. So there are a lot of sources of uncertain bias that come in, which you're only starting to understand. So yeah, Pierre, please take it from here.
Thanks. Okay, here we go. Here it is. I'm sharing the screen. Tell me when you see it, please.
Yeah, we can see it.
It's okay? So the challenge on my side was to estimate tree carbon from tree crown area. So just to introduce, we started by this Sahel landscape, typical Sahel landscape in the dry season. The photo has been taken in the Ferlo region of Senegal. In the middle, you have the view of the very high resolution satellites. So you see all the top of the canopies and the shadow on one side. And on the right hand side, you have the map of the individual trees with the canopy again. And the colors actually are just the classes of size. But we know for each individual tree, its position, latitude, longitude, and with precision, and the area of the crown in square meters. So next for me is this. No, it doesn't work. Okay. So the classical allometry equation by foresters. First, the equations I established per species or group of species. And the challenge was to have equations that were valid for all species found in this area. So the equation classically I established for wood volume, expressed as a function of a linear function of the square of the diameter measured at breast height, as indicated by this this guy on the right hand side, and the height of the tree. So just the equation you have in red in the top. Then to assess wood mass, you just multiply the volume by the wood density. So it was not common in the area to assess the mass of the wool branches plus the wood trunks, and nor even more no common for the leaves and for the roots. We measured it in different places, the Sahel, mostly in Mali and in Niger, as indicated by the green stars in the map in the middle, top. And we tried very much to get access to the existing database on woods by the Forestry Service of different countries in the Sahel, but either they had not measured the crown area, or else they were not ready to share the data. So we end up with our own data that we collected for the International Livestock Research Institute in Mali and Niger. They were interested in essentially the leaf mass, but we also measured the wood mass and the root mass for a few species and a few individuals. In total, we had measured the leaf mass on 900 trees from 27 different species on the left side. For the wood, we had a bit less, 698 trees from 26 species. And for roots, you can imagine it's much more laborious. We had only 26 trees from 5 species. Okay, you can see on this linear regression that the values are quite spread up, and it's expectable from different species. For example, in leaves, you can see that you can make differences between in relation with the size of the leaves, with the micro leaves, the meso leaves, and macro leaves. In the case of wood, you have a difference between the bushes, the shrubs, and the trees, depending if their top are flat, round, or narrow. And for roots, it's very different between bush and shrubs on one side and trees on the other. Hopefully.
So, and the linear regression, because yeah, there's a bunch of lines going. Is each species a clean line? It's kind of hard to say what your variation is.
Well, initially I had for ILRI, I worked with species, so I had but with the classical type of allometry function, with the trunk diameters, for example, at the height of the tree. But here the challenge, we don't know the species with the satellite data. Okay,
Yeah.
so I have to find a way to assess. In this case, that was just mass, the dry mass of leaves or wood or roots, just from the crown area. Okay, so the first plot was the linear, and I was desperate because you could see the functions, even the main ones, so where you have the functions on the top, you can see that the air squares are pretty poor, except for
Right.
but the roots, there are very few data. So now I was
But then what do you do? I mean, in the sense that I know like in US data sets, like the Forest Service collects pretty fine grained plots of what species distribution there are. And it looks like in your data set, you do have cleaner bands within a category, right? So is that a viable approach, just to say the distribution is like 10% of this trees in this area is this species, that species, that species, and have per species plots?
well, we are lucky as Jim said, that in the Sahel, at least from east to west, the flora is quite constant.
Okay.
Okay, but then it differs with the eyesight. So from hyper-arid area to the sub-humid area. So the flora differs, and obviously you have more species in the more humid area than in the drier place.
Right. And so if you were to get a new data set, like would site surveys make sense? Would close-up drone imagery of, that can identify species, would that be usable?
Well, my hope, we'll come to that point at the end.
Okay.
My hope is that with the viewer, we could have a large data set that would help to identify species. And certainly, if we knew the species, will improve on the allometry.
That makes sense. Thank
That's
you.
not the case for the time being.
Right.
We have to go with this, and hopefully, when you log transform on both sides, so the crown area on one side and the mass variable on the other, for leaf on the left-hand side, wood in the middle, and root on the right-hand side, you can see that we improve dramatically the relationship with these power functions. Okay. Then we tried alternative models. So the log transform was one. We tried to see if the Baskerville correction that is advertised for this type of log transform improved or not the relation. Then Ankit actually and tried the nonlinear regression, and we also had the ordinary least square. So for the colors, the plain log is in red. The Baskerville correction is in blue. The purple is the nonlinear regression, and the darker green, the ordinary least square. So we had a bunch of relationship that we tested classically with the fit indicators. So the root mean square, the coefficient of determination, the approximation error, and I already mentioned, well, Ankit already mentioned that the approximation error was additive between leaves, wood, and root. So we could have an approximation error for the tree, for the wool tree estimate, mass estimate. So here I took examples, a few examples, well, just for one. So that's the Baskerville corrected log transformation. You have the RMSE for leaves, for wood, and for roots. The R squares for each of them, quite poor for leaves and wood. And the approximation error, which is in kilo dry matter in each case. Say 0.43 kilos for leaves, 12 kilos for wood, and 6 kilos for roots. So in total, when you aggregate it with this function, you have an estimate of approximation error of 13.8 kilos per individual tree in average. Okay. Then we also looked at on the predictions by these different functions. We look also at the ratios of foliage to wood mass to see if they were plausible. And that was the case, but some of them were quite low. When you increase the crown area, you were getting values that are very low and a bit suspect. We also looked at the root to wood mass ratio, and some of the functions, especially the nonlinear regressions, were completely off in this case. So they were excluded for that reason. Then we compared the allometry that we retained. So the two log ones, with or without Baskerville correction. We compared with the published allometry equations for African tropical trees after they were converted to crown areas, because initially they were not expressed with crown areas. They were expressed with diameter of the trunks generally and height. And we were very lucky to find that our relations were, they are the green ones in these two graphs. They were in the ballpark, let's say. So nothing odd compared to existing or published allometry functions. Now, how to go from dry mass to carbon stock? The carbon stock of each individual tree is inferred from the dry mass by multiplying the dry mass by the mean carbon concentration, which is 0.47. It's really quite narrow, somewhere between 0.46 and 0.49, depending on publications. And it's true for foliage as well as wood, as well as root masses. So now the perspective for improvement of allometry equations. One of the first points, we tested the allometry equation with crown area alone, but we also tried with crown area times height. And it certainly improved the fit for wood and for roots, not for leaves, but for root and roots, which makes sense actually. So the question or the challenge we have for our friends in remote sensing is how to assess tree height of individual woody plants by remote sensing. I think there are progress done recently on that point, but not yet at the scale of the work done in this Nature paper. But probably soon we'll have this, and this will be a way to improve the allometry equation. The second way was to develop allometry equation per species, or at least for a group of species that are a bit alike on physiology, phenology. So again, the challenge is put on the remote sensing. Is deep learning able to identify a species or group of species? Again, we have already answers there for, I would say, a couple of very special trees and shrubs. There's a paper published by one colleague in Copenhagen University, Ting Ting Lu, her name, the first author, that was able to map with the same techniques the Federbia albida, so one of the particular species. Another one candidate is the baobab tree, Adansonia digitata. But I mean, soon this could be generalized to other species. Then a third way is to use more field data, because as you've seen, we have used very few of those data, especially for roots. So this will require wide collaboration to build up a woody plant database. And I think the viewer published at the same time than the paper could help in this prospect, to gather people in the forestry services and get this woody plant database built up. Thanks a lot. Oh, the last one. That's for Jim Tucker. So the photo of all the, or at least most of them, there's a few missing.
So cool, yeah. Thank you very much. I guess one thing I wanted to elaborate on is the utility of this for understanding kind of evolution of forests. So evolution of forests as a result of fire, logging, beetle infestation, things like this, response to stimuli, because you do have these time series of forest observations.
Yes, that would be very easily done. We were forced to use what imagery we had on hand. We actually started this work in mid-2020, and at that time we only had 326,000 images. Now we're up to 800,000 images for the area, and we will at least have 2 time periods. But we demonstrate the potential if you had more recent data and a focused objective. You could do very, very well if you have the data.
And in terms of your satellite, the ability, so you have 50 centimeter data from your satellite. How fine grained do you truly need, and how the time gaps between images? How wide can it be before it's useless?
Well, you saw the figure in my presentation that 50% of our trees come from a canopy diameter which is small, where the canopy area is 15 square meters or less.
Right.
So, for example, if you use Planet Lab data with a 3 or 4 meter spatial resolution, 15 square meters is about a canopy diameter, assuming a circular crown, of about 4 meters. And so those would be very difficult to map with 4 meter data. And so it depends upon the satellite data you have. And Maxar 50 centimeter and 30 centimeter data are what we prefer to use because of the spatial detail.
So are there any other sources like Maxar that go farther back in time or cover gaps in time and winter?
Unfortunately not. There may be in the future. So all of our students may be able to do this much better in a few years. But at the moment, not, or no is the question, unfortunately, or no is the answer, unfortunately.
Right. And then, so I can see how in these dry climates, tree cover is easier to pull apart. And we were talking about denser canopies. So pulling this out over the rest of the world, I'm thinking, pine forest, like would be relevant for forestry, or like jungles for deforestation. Like how far and what precision is necessary to map the world's forest?
I would think with 30 centimeter data. Now, the reason I say 30 centimeter data is because that below 30 centimeter, all data are classified and cannot be distributed or used or sold commercially. But at 30 centimeters, you could do a much better job in closed canopy forests. And this is something which Ankit and our collaborators in Copenhagen are working actively on. And I personally think that there's reason to be optimistic that they will work out a method with machine learning to map the tree crown diameters in contiguous areas of forest.
Excellent. And what about things like grassland, bushes, soil, which is the nastiest one?
We have no problem with bushes. In fact, we're about to map all the bushes as they encroach into the tundra as conditions warm. And we actually, in our study, mapped 1.5 billion bushes of canopy area from 1 square meter of area up to 2.75 square meters of area. So we actually have those data, but as was mentioned by Ankit, we have an increasing number of omission-commission errors as we go to smaller and smaller canopy areas below 3 square meters.
So, okay, the objects you're looking for are like 3 by 3 meters, roughly. I mean, this in the smallest end. That's about the size of a, like a shack. So it seems like the same approach can be used to map human populations down to like small shack level.
Well, we can easily, I think we may, when we do this again, go down to 2 square meters of canopy area because we have the data now and we have methods to minimize the confounding effects of areas of trees and bushes. You have to realize this paper is a first paper and we're working actively on improving several aspects of it. And that is one of the things we're looking at, is to go to smaller diameter trees with their associated shadows.
Okay, and that should apply to buildings and whatnot. Oh, and speaking of which, actually, you mentioned you focused, I think I caught this, that you were removing the off-angle images and we're focusing on the mostly down images for consistency, right?
That is correct.
If you specifically focus on off-angle images, your shadows are going to be a lot more obvious and heights are going to be more obvious. Is that useful?
Yes. Yep. And you have a great distribution of different viewing perspectives. If in our paper, in the supplemental information, we have plots for every one of our UTM zones, all 10 of them, in terms of the off-nadir angles, which were used in our studies, and we could very easily change our selection process to only use off-nadir images if that was what you needed to get the results you needed.
All right, awesome. Yeah, that sounds very promising. Well, thank you very much. This is like awesome, fascinating work.
Well, if there's any possible collaboration with Google on some of the things we're doing, we would welcome it. We're just starting. We ended up, not surprisingly, with more questions than answers. And we also have access to all of the Maxar satellite data. And we could very easily have a project together, and we would welcome working with the people at Google because of your expertise in related areas that are very important to us.
Yeah, that'd be awesome. Hopefully that will help, some folks in the audience will be like able to help with that. But yeah, thank you.
Thank you.
Yeah, thanks. Bye-bye.
Bye.
Okay, bye everyone. And thanks a lot, Greg.
Absolutely. Thank you.